The overall goal of this new isolation strategy is to widen the number of new virus isolates in order to study the pangenome of this new viral family and to better understand their ecosystems and potential pathogenicity to humans.
This method can answer key questions in the virology field to help virologists worldwide interested in prospecting giant viruses and Faustoviruses in particular.
The main advantage of this technique is that it shows new improvements through many stages, like the antibiotic and antifungal mixture, the new starvation medium adapted for the amoeba, and the use of flow cytometry for faster and more sensitive disease detection.
After collecting dirty water samples according to the text protocol, prepare the host cell V. vermiformis by adding the amoebae to a 75 square centimeter cell culture flask with 30 milliliters of protease peptone yeast extract glucose or PYG medium, and incubate at 28 degrees Celsius for 48 hours. Use counting slides to count the amoebae, and when they reach a concentration of 5 times 10 to the 5th to 10 to the 6th cells per milliliter, harvest the cells by centrifugation at 720 times G for 10 minutes. Aspirate the supernatant and use 30 milliliters of sterile Page's amoebal saline or PAS to resuspend the amoebae pellet. After rinsing the pellet a second time, use 30 milliliters of starvation medium containing an antibiotic and an antifungal to resuspend the cells to approximately 10 to the 6th amoeba per milliliter. Starvation medium enables the amoebae to stay alive without encystment.